Okay, Bismillah ar-Rahman ar-Rahim. And I don't want to encroach on Dr. Deeba's time, but she told me that you guys might have questions on the one lecture I gave at the beginning of the week. So I will be here for 10 minutes. Uh, so if you have any questions on the lecture, let me know. And I don't have any prepared MCQs on the material I gave this week. في أي سؤال؟ Any questions? No? Okay. Yes. كله داخل معكم. اه الريجوليشن هاي. لا ما قلت هذا الحكي أنا في الامتحان. This lecture is not part of the midterm exam. The post-translational modification is with you, except for the last slide. Yeah. And I stopped at phosphorylation. فالphosphorylation is with you. بس الجلايكوسيليشن وبعدين الكاربوكسيليشن والبيوتينيليشن هذا is not part of the midterm exam. فما في 20 أو 30 سلايد. من وين جبتوا الحكي هذا؟ ما always get the words from the lion's mouth زي ما بيحكوا. هي الليكشر كلها أصلا 20 سلايد. وهون for the regulation يعني everything is with you. يعني text I did not go over the entire text. يعني there are some slides that I did not read. But this is for you to know and study. Okay. ف... ها مش بالمتين بس راح أكون معكم بالفاينل. yeah. كيف يعني؟ هلا يون ما راح تحفظ ما راح تحفظ الجينيتك كود، بس حكيت تحفظوا الستارت كودون والستوب كودونز and how to use the genetic code in order to figure out the amino acid. I don't think Watson or Crick themselves know the genetic code by heart. ف okay, any questions on regulation of gene expression? محاضرة الترانزليشن؟ لا هاي this was last week's tutorial. ممكن you can come to the office hours. ها تفضلي. correct. activate or inactivate protein. an already activated protein مثلا. So it is an on off switch. تمام. هلا when you turn on the lights you turn them on. فimagine عندك إنه you phosphorylate a protein, you activate it by phosphorylating it with a kinase. هلا مش to inactivate it, to bring it back to the basal state you remove the phosphate group. تمام. بس يعني phosphorylation does not necessarily mean activation برضو. You need to put this into mind. Sometimes it is the other way around. يعني ها لا لا مو شرط. ممكن يكون بروتين يصير له phosphorylation يصير inactive. you remove the phosphate group بترجعه للactive state. فit depends يعني on the protein. يعني you will get exposed to this a lot during the second semester when you study biochemistry. يعني this is a quick way by which the cell regulates the activity of proteins. Okay, by kinases و phosphatases عشان it is a simple يعني biochemical reaction و rapid. تمام. Okay, no questions on the regulation عشان خلاص يعني this will be my first my last tutorial. Okay, and I will leave the floor to Dr. Deeba. So again يعني you can come to my office hours anytime. You can come to the office anytime. If I'm busy I will let you know that I'm busy. بس يعني I will try to answer your question. على الريجوليشن مش داخله في الامتحان يا بنتي في الميد تيرم. حق الفاينل لا I did not prepare any MCQs. Yeah.
Now you have already taken the resource just yesterday and today, so I will just skim through it. So it was about protein transport, and we discussed about the two different types of ribosomes, free and bound, and we talked about how proteins that are produced by the bound ribosomes, where do they end up in? So they end up in membrane of the ER, or in the Golgi, or outside the cell, or in the lysosome. So how does it work? First of all ribosomes are free. As soon as one microRNA mRNA comes, it binds, it makes a protein, and that protein has an inbuilt what? Signal sequence. That signal sequence can be recognized by a big protein in the cytosol, which is called as SRP. This SRP binds to it, takes the whole complex to bind to a receptor on the membrane of the ER near an opening, and that opening is called as translocation complex. Now once the whole complex is bound, in ribosome is constantly translating the protein. That protein enters into the lumen, and once the translation is over, the ribosome, it dissociates and the protein moves inside the lumen. The signal peptide also gets removed. Now when the protein is getting synthesized and entering into the lumen, at the same time modification happens, and the most common modification is adding of sugars. The sugars are initially attached to a lipid molecule dolicol, and as the protein is entering the lumen, the whole bunch of sugars get transferred to a sequence which is called as SNXer or SNXther to the SN part of it. And because it is getting transferred to the SN, it is called as N-glycosylation. Similar glycosylation happens in the Golgi also, but on the SER and THER, and that is called as O-linked glycosylation. If you have any questions, you can stop me. Now, once the protein have been formed and modified in the ER, it can stay with the ER or it can move towards the Golgi, and when it is moving towards the Golgi, the membrane of the ER forms a bubble around it or a vesicle around it, and then the protein is diffused. When the vesicle diffuses with the cis Golgi, the protein is inserted. However, this does not happen randomly. The carrier vesicle has a coating of a protein called as V-SNAR, and the cis Golgi in a specific region has a coating of T-SNAR. So, wherever these two proteins are there, only then that fusion and insertion of the transported protein takes place. As the protein reaches the Golgi, it goes through different stacks of the Golgi which are filled with enzymes that carries out the modification depending on the destination of the protein. For example, if it is going to lysosomes, what is added? What is added? The phosphate is added in the Golgi. So, the tag is mannose-6 phosphate. So, any protein or any enzyme that has a mannose-6 phosphate, it will go to the lysosomes. And the other modification I have already told you, O-linked glycosylation can happen. Once all the modification has happened, the different proteins go to different regions of the trans Golgi, and there they are sorted and get enclosed in a vesicle, and they go to different destinations. For lysosome, the acid hydrolases that are synthesized by ribosomes, I'll just go on this slide, that are synthesized by the ribosomes attached on the ER. The protein gets a mannose in the ER itself, and those acid hydrolases, they come to the Golgi. In the Golgi, the enzymes add a phosphate on the mannose, so it becomes mannose-6 phosphate tag. And all those enzymes then in the trans Golgi, they go to a specific region and bind there to the receptor. The receptor only recognizes mannose-6 phosphate. Now, when all the enzymes are bound to the receptor, the membrane bulges, forming a vesicle, and a protein coating is needed for vesicle formation. That protein is clathrin. And once the vesicle has been formed, the clathrin coat gets removed, and as the vesicle is moving in the cell, at the same time whichever protein or an old organelle needs to be digested, it gets enclosed in a vesicle which is called as endosome, and then this vesicle carrying the acid hydrolases combines with the endosome collectively, forming a primary lysosome which is inactive. Now, to activate it, hydrogen ions needs to be pumped inside this endosome or the primary lysosome. As the protons are pumped inside, the pH becomes acidic, and when the pH becomes acidic, the enzymes get released from the receptor, the phosphate gets released, the enzymes become active, they do their function of breaking down the whatever biomolecule is here. Yes. Yeah, it is for any vesicle that is coming out of trans Golgi. Also, for the secretion of proteins outside the cell, there are two mechanisms. One is called as structural or constitutive mechanism. This mechanism of secretion does not need any stimulus on the cell. And usually it happens for the proteins which are constantly needed by the outside environment of the cell, for example housekeeping proteins. However, the regulatory secretory pathway is for those proteins which are only needed when some mechanism is happening. For example, digestive enzymes or hormones, they get secreted only when there is some stimulus for them, and we had discussed about insulin, we had discussed about salivary enzymes. They are secreted only when food is there in the mouth, or when the blood glucose levels are high, only then insulin will get secreted. Trafficking of proteins synthesized in by free ribosomes. They either stay in the cytosol or they go to nucleus, mitochondria or peroxisome. They do not have an inbuilt signal sequence in the N terminal, but they do get tagged by other sequences, other structures to help them go inside these organelles. Cytosolic proteins, they do not have any form of signal on them, neither an N-terminal signal or any special structure on them. As a result, they stay in the cytosol. For all those proteins that are synthesized by free ribosomes, a signal is added later on. Okay? For nuclear protein, the proteins are formed and then a signal is added to them called as nuclear localization signal. Once this signal has been added, this signal is able to bind to a transporter protein called as important alpha and important beta. Now important alpha first binds to the signal and important beta binds to the alpha, and important beta itself is attached to an energy molecule which is GTP. The whole complex then enters inside the nucleus, and once inside the nucleus, the GTP breaks down to GDP. As a result, the important protein also gets removed from the nuclear protein, and important proteins, they come out to bind to new nuclear proteins to help them enter the nucleus. Mitochondrial proteins, they are big proteins, so they need to be linear with chaperones added, and to keep them linear, chaperones are added to them, chaperone proteins, which also require ATP for their job. Once these proteins are in their linear form, a signal sequence is added to their N terminal, which is called as import sequence. Again, this is added, not a part of the protein. Once it is added, the import sequence binds to a receptor. First, it binds to a receptor that is close to a complex called as TOM. Complex is simply an opening in the membrane, and then the protein enters into the intermembranous stage. Once it is into the intermembranous space, the import sequence again binds to a receptor that is present in the TIM region, that is in the opening in the inner membrane, and then finally the protein goes inside. Once it is inside, it gets folded and the signal sequence gets removed, and now the mitochondrial protein is fully functional. And finally, peroxisomes, it is transported in a fully folded and functional form. It does not have any sequence attached to the N terminal, rather it has a sequence at the carboxylic group, carboxylic end, which is called as peroxisomal targeting sequence. With the help of this sequence, the peroxisome protein binds to the receptor, and once it binds to the peroxisomal receptor, the receptor carries the protein and it sits on the membrane of the peroxisome, and now the protein can enter inside the peroxisome. Again, the signal that is there, it does not get removed. Okay. This was all. Any questions? So I have two of the questions as sample. Let me see if you are able to answer them. Protein with a mannose 6-phosphate tag are destined for? Very good. Lysosomes. I will put them on the Moodle with the answer. Which of the following is correct about membrane bound ribosomes? What will be the correct answer? They synthesize proteins which become part of organelles or cytosol. Read again, it is membrane bound ribosomes. Membrane bound ribosomes. Those that bind to the ER. B is the correct answer. Proteins synthesized by them become part of any of the membranes. Okay? It can become part of ER membrane, it can become part of Golgi, it can become part of cell membrane. And it is also mentioned in the text that they get associated with membranes. Okay? I'll put this on the Moodle with the answer.